identified this region, homozygosity, so this region that looked the same in all the patients that were affected and different in the patients that were not affected. And in that region, among the many genes, was one gene that's called Megalin. This is the gene product, so the protein of that gene, and that is mutated. And by sequencing that gene, she found mutations. She found the actual mutation that causes the disease. So since we said this is recessive, mutations in both copies of the LRP2 Megalin gene were identified in all members of the consanguineous kindred that were affected. Healthy parents had only one mutated copy of the gene, so this is consistent with what they had observed, clinicians had observed before about it being recessive. And also, when we looked, not only in the consanguineous families, but all the kids in the world that have Donnai-Barrow syndrome, we couldn't do homozygosity mapping on them. But looking at the single genes, we found that they also had mutations in that gene. So you can see how a rare family, a rare consanguineous family, can actually point to a gene that then is helpful also for diagnosing many other kids. Megalin gives us a very important lesson in fetal development, because DBS abnormalities occur in very different organisms, in very different organs. We said the eye, the ear, the brain, and the pattern of abnormalities that we see in patients that have Donnai-Barrow is strikingly similar to the pattern of expression of that gene. So if we look at mouse embryos, or if we look at humans, that gene is turned on during fetal development right in the organs where the disease will strike the most. So that gene is expressed in the developing brain and spinal cord, it's expressed in the developing eye, it's expressed in the developing ear, and it's expressed in the developing kidney. It's probably not expressed in the diaphragm. That can, that can confuse us a little bit. But actually, no, because it gives us another mechanism and how to look at how diaphragm develops. Probably the heat that hits the diaphragm comes through the kidney or through the lung, because the gene is also expressed in the lung. So Megalin functions as a cellular receptor. So it's a protein that sits on the membrane outside that cell, and its job is to take certain things that are outside and bring them inside. Some of those molecules that it does bring inside might be the ones that are responsible for CDH or other characteristics of DBS. For instance, Megalin also works on getting vitamin A when it's bound to the vitamin A binding protein and bring it inside. And vitamin A is probably very important in modulating diaphragm growth. And this we know also because of some earlier mouse studies and other animal studies that fed a particular chemical to the mice. So currently we are submitting a study protocol that aims at identifying possible areas of interventions in these Donnai-Barrow patients. So allowing us to possibly see if it's possible to detect early complications and maybe supplement the things that this Megalin has a hard time reuptaking and loses in the urine altogether. And see maybe if that can help other things. So the conclusion is the bottom line. That the study of rare genetic disorders is really important in genetics, not only for the disorder itself, so for the patients that have that specific disorder, but really because it opens up our mind to several different pathways, several different mechanisms that actually enhance our knowledge a lot on also isolated cases of CDH. And that's it. And the acknowledgements are the same as yours. So you can read those.
You
should acknowledge the same people.
Wake up, or are we okay? I don't know.
Anybody have a specific question?
We can do it later.
You could have, it doesn't matter. You can for me, you can ask questions during the time, just so you don't, if there's any confusion, I can clear it up at the time I'm saying it. So, but.
I did just have one quick question.
Yeah.
I've been asking a lot of questions. The CDH and CDH plus, or isolated CDH and CDH plus.
Plus other things.
Yeah. There are some other things, and probably that, you know, that may be considered like normal variants. Where do you actually draw the line? You know, how serious does the plus have to be for it to be?
Some pluses are not real pluses, like some kind of intestinal malformations or heart malformations are not considered to be actual pluses because they are consequences, or for instance lung hypoplasia, because they are actual consequences of the diaphragm missing. So those really don't add up to the plus thing.
What about CDH and ASD?
That also I wouldn't consider CDH.
Okay.
Although it's very hard to say when you split the heart phenotype in its different possibilities, different abnormalities you can find. It's hard to say which is primarily bound, which is primarily bound. Plus, not all CDH pluses are syndromes. Some of them are associations. So they've not been observed enough times to really say it's a syndrome. But when you say it's a syndrome, that's a word that comes from the Greek. I think we discussed it last year. It means running together syndrome. So they need to have possibly the same cause to all fit in a syndrome. Otherwise, there can be associations, or there can be sequences, or.
Okay.
Like scoliosis could be associated because of the patch, not necessarily congenital.
That's congenital, though. The plus is only congenital. So whatever happens later because of a surgical intervention or because of things that form later are usually not pluses, except there's something that happened independently. Like the myopia that we see in the Meaghan patients is progressive. It goes worse over time. So that's still a plus.
So even though she had a PDA and an ASD, so even though those are defects, those could be just associated with, that would make it a plus.
Yeah.
Okay.
So thanks for having us here. It's great to be here and meet you guys. We haven't met a bunch of you yet, but hopefully we'll get to. But I'm going to talk about a little bit of a few different things about our study, as well as how we're trying to understand the etiology of CDH, or the origin of CDH in our laboratory. And feel free if you have any questions during, you can interrupt me. So these are some pictures of some of the kids that we've enrolled into the study, and they're published in our newsletters. So I brought our next version of our newsletter that we just finally finished last week. So I have a pile of them if anybody wants one. And we have previous newsletters as well that I can email you or send if you want to read them. It's just different stories from families and just some of our research at different points during our study. So our study aims to determine which genes or gene pathways that when they're perturbed or when they're not functioning properly lead to CDH and small or hypoplastic lungs. We hope that in the future, which is far future most likely, but we'd really like to design effective but safe drugs that can treat babies in utero to help enhance lung growth before birth. And we also hope to improve care and outcomes for the next generation of babies with CDH. And I guess that's sort of where we're starting with our Denai-Barrow syndrome work, is we're trying to use interventions just for that small group of kids, but at least it'll help them because it's a progressive disease later in life. So we're starting there, but I mean, this is a sort of a general, we hope to help. You know, if we can figure out the, what everyone else is trying to figure out.